Jess Eaton from Carnegie Mellon University, and I will let you say your title.
Sure. All right, so just to let you know, this talk marks switching over to eukaryotes from prokaryotes. So hello, everyone. I'm Jesse Eaton. I was a student in Professor Russell Schwartz's lab at Carnegie Mellon in Pittsburgh, studying computational biology. I'll be presenting my work from my master's. So understanding the composition of a patient's tumor is becoming increasingly important as personalized treatments for cancer become more prevalent. One of the major challenges in characterizing an individual's tumor comes in the form of a noisy raw signal. When the tumor of a patient is completely sequenced, the sequence is really just a mixture of reads that could align to any of the millions of cells that make up a tumor. So there are many tools out there that, given a tumor's genomic data, will try to both infer what cell types the tumor is composed of and the phylogenetic relations between the cell types. So we know a tumor is originally comes from a single cell that clonally expands. Over time, one of those clones can acquire another mutation, and if that mutation is advantageous for the tumor, it will also clonally expand. And we can model this process as a phylogenetic tree, where nodes are clones or cell types, and edges are mutations. There are a few types of mutations that can occur, and our method focuses only on a subset of those, so I'll quickly go through the different mutation types. So single nucleotide variant, or SNVs, are point mutations, where single nucleotide is changed from one nucleotide to another. Copy number variants, or CNVs, can be duplications, depicted here where a segment of DNA is copied and both copies are kept in the genome. Copy number variants can also be deletions, where a segment is deleted from the genome. And structural variants, or SVs, can be where a segment is moved from one position in the genome to another. So our method focuses more on the copy number variants and the structural variants. So I'll explicitly define what I mean by a structural variant. We define a structural variant as a pair of breakpoints found adjacent to one another in the cancer genome, but not in the original reference genome. So in this example of a tandem duplication, we can picture the individual boxes as nucleotides and the similarly colored nucleotides as segments of the genome. If segment 2 is tandemly duplicated, then the nucleotides originally at positions B and C are now adjacent to one another. So we say nucleotides C and B are a mated pair of breakpoints. Similarly, if you have a deletion of segment 2, now nucleotides A and D are adjacent to one another. Now that I've defined what I mean by a structural variant, I'll give you the goals of this research. So we develop an algorithm that identifies different cell types in a tumor, builds a likely tree that explains how these cell types are related, and assigns structural variants to each of them. So luckily, Professor Jianma's group at Carnegie Mellon, and specifically his student Yang Li, has developed a tool called Weaver that takes as input called reads and uses a Markov random field to estimate the copy number of both segments across the genome as well as copy number of the structural variants. So if a tumor has one or more spatial samples taken from it, then we can run Weaver and get estimates for the copy numbers of variants for each of those samples. So what this means for us is that we can build a matrix that describes each sample as a row. For each column, we have a single variant, with the first L columns corresponding to the copy numbers of breakpoints, and the next R columns corresponding to the copy numbers of segments. We can similarly know from the output of Weaver which breakpoints are mated pairs, which we encode as some binary matrix, as well as what segments each breakpoint originally belonged to. We can do this since each breakpoint describes exactly one position in the reference genome, and each position can only be found at one segment. So with this input, what we try to do is decompose the original matrix F into two matrices, U representing the composition of clones for each of the spatial samples, and another matrix C representing the copy number of variants belonging to each of those clones. For matrix U, each row represents a single sample, and the values are the percent of each clone that makes up that sample. Rows in matrix C hold the copy number profile for each of the variants, for each of the n clones. And when you multiply U and C together, you get the same dimensions as our input F. So this is an example of a simple phylogeny that shows what the output copy number matrix C would look like. If we start with the normal cell type as the root and assume that the normal cell type is diploid, and two mutations occur, one duplication along the edge to the left and one deletion along the edge to the right, then we would get this following copy number matrix. We see the root node, which we put at the bottom row of the matrix, has no breakpoints here and is entirely diploid. So the copy number for each segment is 2. The left child is represented by the first row, where two mated breakpoints have appeared, and the copy number of their corresponding segments have incremented to 3. The right child is represented by the second row, where two different mated breakpoints have appeared, and the corresponding segments have decremented to 1. And the matrix representation of this phylogeny would look like this matrix E, where in the bottom row we see the root has edges to both of its children. So one approach would be to find some U and some C that get as close as possible to the original matrix F. However, even if we solve this, we won't get a model for the relation between different cell types. Also, as you'll see later, we can achieve better results by making some assumptions about the phylogenetic history, as well as some assumptions about the behavior of structural variants. So we add two regularization terms, one that adds a phylogenetic cost, and another that constrains structural variants to remain consistent with their corresponding segments. And I'll get to these terms in a few minutes. One general approach to solving this problem was introduced by Simone Zachariah and Mohammed El-Kabir from Ben Raphael's lab in Princeton. In this approach, one randomly chooses some matrix U, then looping until convergence, you solve a mixed integer linear program for copy number matrix C, and then for copy number matrix C and tree E, and then hold those two constant and solve for matrix U. And this process continues until the objective function is below some threshold or a maximum number of iterations is reached. So we first constrain the model by imposing a phylogeny over the clones and penalize changing the copy number of segments by adding a cost proportional to the L1 distance across all edges of the phylogeny. In this toy example, the phylogenetic cost would be 4, since the duplication to the left child spans 2 segments, and the deletion to the right child also spans 2 segments. So now that we've constrained the clones to form a phylogeny, we can start assigning structural variants to nodes. So one way to do this would be to make the infinite sites assumption. This assumption is usually made on SNVs and makes the assumption that the genome is sort of infinitely long, or so long that it's pretty unlikely for a mutation to occur at any single point, and it's highly unlikely that two-point mutations would occur at exactly the same nucleotide on multiple edges in the tree. We make this assumption for structural variants. Because a breakpoint occurs with a single nucleotide resolution, we can make the assumption that any exact segment is unlikely to produce a structural variant across multiple edges of the phylogeny. So we allow the copy number of each mated breakpoint pair to increase from 0 to 1 exactly once, but can subsequently be lost. Of course, if it decrements down to 0 sometime later in the tree, then it's unable to increase at all because the chromatin is now lost. One thing I'd also like to mention is that we have linear constraints for each of these, but I didn't want to clutter the slides with formulas, so please see our paper if you're interested in those constraints. So now, to relate the structural variants to one another, we apply an ancestry condition, saying that some structural variants S, that if some structural variant S appears before some structural variant T, and S is never subsequently lost, then the fraction of clones containing S should be larger than those containing T. We apply one last condition that attempts to keep each structural variant copy number consistent with the copy number of the segment it belongs to. So this is done by pre-calculating the ratio between the breakpoint's copy number and the copy number of the segment that that breakpoint resides in, and adding in a penalty for any inferred breakpoint that deviates from this ratio. To give an example of why this might be important, let's suppose we had two different scenarios, a tandem duplication and deletion. In this situation, in the situation of the duplication, the copy number of the breakpoint, let's say at position C, increases to 1. And the copy number of the segment that contains the breakpoint increases to 2, as seen by the two blue segments. And their ratio is 1/2. In the situation of the deletion, when segment 2 is deleted, we look at breakpoint A. And we see that the copy number of this breakpoint is 1. But the copy number of the segment containing that breakpoint is also 1, making their ratio 1. So forcing the copy number of breakpoints to remain consistent across nodes helps maintain the distinction between these two different copy number variant types. So for this, we simulated data. We generated, on average, 50 mutations, leading to approximately 100 breakpoints across 3 chromosomes. We randomly generate some matrix U and combine that with the copy number matrix to create the input for our algorithm. We first ran a simulation without using any regularization terms, depicted as this red line. Upon adding these terms, we see a significant increase in the closeness to the original copy number. Here, lower is better, as the y-axis is the L1 distance between the inferred and true copy number matrices. We originally performed a grid search across hyperparameters and found that problems of different sizes had different optimal parameters, meaning more or fewer structural variants. We then decided to automatically select the hyperparameters based on the number of samples, segments, structural variants, and clones. This was done in a way so that the error for each of the terms in the objective function were equivalent. We found that automatically setting these parameters performed equally well as doing a grid search. So following simulation, we evaluated our algorithm on 59 TCGA samples for which whole genome data was available. In general, these samples had a large variance in the number of structural variants, ranging anywhere between 4 and 350 breakpoints called by Weaver. We found that there was a diverse population of phylogenies from the various tumor samples. None of the topologies were found to be purely linear, which is consistent with a model of subclonal heterogeneity. The phylogenies inferred from the TCGA patients contained a diverse set of unique clones ranging between 4 and our allowed maximum of 7. We also see a bimodal distribution, which is suggestive of the fact that there could possibly be 2 mechanisms generating the structural variants. So to conclude this talk, we ultimately found that adding phylogenetic constraints, as well as the consistency between the structural variants and their copy number segments, increased performance in estimating a copy number matrix. We also found significant diversity among the TCGA patients. Some room for improvement for this method include reducing the time it takes to run. On average, the TCGA samples take around a day to process, but others with larger number of structural variants were unable to finish in the specified time limit. The algorithm also requires a substantial amount of memory, making many of the samples with a significantly large number of structural variants difficult to process. So all this work was done in Python and is openly available on GitHub. I'd lastly just like to give thanks to everyone who's helped me with this project. Russell Schwartz, my advisor, gave me a lot of great advice along the way. I'd also like to give a special thanks to my co-author, Jingyi Wang, who worked in Jianma's lab. So thanks a lot to everyone.
So thank you a lot. So we have time for 2 or 3 questions.
Yeah.
So I'm curious about your rooting. Do you also include healthy tissue sampled elsewhere? And in that context, when you sequence a tumor, you're getting normal cells near the margin or any
Right.
angiogenesis, invasion or something. How do you deal with that?
Right. So the short answer is we haven't dealt with that. We do make the assumption that the root node is completely diploid. But also we know that the sample actually comes with normal contamination. So that's where we allow a certain percentage of the cell types to belong to that root node. So we don't just restrict it to like only the leaves get a % of the cells. We can also say, okay, there's 30% contamination or something like that. Yeah.
Jesse,
Yes.
very, very clear talk. Thanks.
Thanks.
What were the T-series samples you actually ran? What cancer types?
They were breast cancer samples.
Breast cancer. Okay.
Yes.
And in terms of a little bit more to that last question, then,
Yeah.
the heterogeneity of other cell types that contribute, especially in cells like pancreas, where there's a lot of stromal reaction. I wonder how a model like this would perform, or an approach like this would perform. And additionally, the assumption you make on assuming that a prior, earlier occurring mutation is going to be more prevalent in the population doesn't account for the out competition or cellular death or that kind of thing, which is very frequent in cancer. So I wonder how you address that and think about that.
Right. Okay. So that was, I guess, 2 questions. So one being, how do we account for different cell lineages? And as of now, we don't. We could maybe add in some extra nodes that are not attached to the phylogeny, but right now haven't added that into the model yet. And then, sorry, could you remind me of the second question?
Cell death and the assumption that you make with
Right.
there having to be more of an earlier.
Right. So we only apply that assumption if the subsequent clones do not lose the mutation. So if the mutation is retained in all of the children or in all of the descendants, then we apply that condition. Otherwise we just don't apply the condition. Yes.
Yeah, last one.
So could you apply this to inversions or fusions in translocations? Because you are just taking into account breaking points, right? Could you use for these settings?
Right. It could. We have not taken a look at translocations yet. I mean, they are in the data set, but we're not doing anything specific about the model to account for those. But that is a good area of future work.
Okay. So thank
Right.
you again.
Thanks a lot.